Welcome to Forward Fest. We are so glad you are here engaging in some bold ideas, varied approaches, and forward thinking that make Princeton the special place that it is. We want to hear from you throughout a year of forward thinking, and particularly during Forward Fest. Be sure to tag any of your forward thinking posts on social media with hashtag Princeton Forward. That includes any questions or comments you want to share with the Princetonians who will be broadcast today. It is my pleasure to introduce you to the moderator of our Thinking Forward bioengineering session, Kirsten Bibbins-Domingo, Princeton class of 1987. Dr. Bibbins-Domingo is the Lee Goldman, MD Endowed Chair in Medicine, Professor and Chair of the Department of Epidemiology and Biostatistics, and Vice Dean for Population Health and Health Equity at the School of Medicine at the University of California, San Francisco. Today, she will be leading a thoughtful discussion with 4 of Princeton's forward thinkers, cutting across departments and disciplines to make a positive impact on health, medicine, and quality of life.
Thanks, Sophie. Welcome to Princeton University's Forward Fest and today's session, Thinking Forward Bioengineering. My name is Kirsten Bibbins-Domingo of the great Princeton class of 1987, and I am delighted to be with you today. I'm looking forward to digging deeper into the exciting field of bioengineering, and I am really thrilled to be joined by 4 Princetonians who are forward thinkers in this area. With us today are José Avalos, Assistant Professor of Chemical and Biological Engineering and the Andlinger Center for Energy and the Environment, Cliff Brangwynne, the June K. Wu, 92, Professor of Chemical and Biological Engineering and Director of the Princeton Bioengineering Initiative, Celeste Wilson, Wilke Family Professor in Bioengineering, Professor of Chemical and Biological Engineering, and Director of the Program in Engineering Biology, and Ben Raphael, Professor of Computer Science. We'll start by welcoming Cliff Brangwynne to the discussion.
Thank you, Kirsten, so much for moderating this session. I'm really excited to be here and share some of the excitement at Princeton with all of our audience members and join this discussion. Yeah, I'll share a little bit about why I'm particularly excited about bioengineering at Princeton. I think bioengineering is so exciting because, especially at Princeton, because it brings together 2 areas of historical strength at Princeton, which is engineering and the life sciences. It's the intersection of those 2 areas. Within engineering, Princeton has a long tradition here. There's 29% of our current undergraduates major in engineering, which is, I think, just an incredible number. Princeton, I believe, is unique in the historical and ongoing embrace of engineering within a liberal arts education. I think that's something that is special at Princeton. And among the students, there is great demand for biological engineering. The students are excited about this. They see this as poised to have the biggest impact of perhaps any other discipline on society in the coming decades. And there's a great need for developing new courses and infrastructure and programming for the student demand. The other part of it is that bioengineering, many people think, and I would certainly agree, that bioengineering is at a place in its development where computers were in the 1960s and 70s. In other words, we're about to enter, we're on the cusp of really the next revolution. And this is an opportunity for Princeton that I think is just tremendous.
Yeah, it's really exciting, and I know that that the excitement is really palpable when we've heard different faculty members talk, and certainly you're highlighting how much this is part of Princeton anyway, in the interest of our students. I'm sitting at a medical school though, and we're excited about bioengineering, but there is no medical school at Princeton. Tell me about why Princeton is the right place for this to happen.
Well, I think that, this question comes up a lot in these discussions. I think that there are many examples of highly successful bioengineering programs at institutes that don't have medical schools. I mean, Berkeley, Caltech, MIT are doing fantastic bioengineering. So there's many examples. Certainly in the applications, some of the most important applications of bioengineering in human health, I would say, they are not the only applications. We'll hear today from José Avalos, whose work in cell engineering for harnessing cells for making chemicals and biofuels and products to benefit society. But certainly, human health is a central focus and a central opportunity for bioengineering. It can be done at Princeton. It can be done with these other examples. It's already very clear at institutions that don't have medical schools. We certainly need to think about partnerships with medical schools. Princeton actually already has some existing infrastructure in the form of an MD PhD program with the Rutgers Robert Wood Johnson Medical School. We're in discussions to amplify that program and tie it into the bioengineering efforts. There are seminar series with clinicians, a recent program started by Assistant Professor Daniel Cohen, and really the list goes on and on. This is something that there's already infrastructure in place, and we intend to dramatically amplify that in our efforts to bring bioengineering to bear on pressing human health needs.
Your own work is really extraordinary and helping us to understand the structures within cells that really are important for understanding some of the diseases that really are major scourges in human existence right now. Tell us a little bit about your own work.
Yeah, I'm happy to do that. I mean, I'm obviously pretty excited about the work that my lab's doing. I think it is a good example, though. I think of the kinds of impact that bioengineering research can have, and especially at a place like Princeton, where there is an emphasis on deep scholarship and fundamental knowledge, even within the engineering school. We're not interested in making widgets that we don't quite understand how they work, but as long as they kind of work, it's fine. At Princeton, there's a deep scholarship and a deep intellectual inquiry. In my work, we've discovered that biomolecules within cells, the building blocks of life, DNA, protein, RNA, come together in ways that are governed by the same types of physical principles that govern how H2O molecules get together into liquid droplets. It happens to be raining here at Princeton right now, or condensation on a cold glass surface in the summer. These are all examples of phase transitions, and biomolecules undergo those same types of phase transitions. What's exciting about this research area is it was really pioneered here at Princeton. There are dozens of faculty on campus whose work is very strongly focused on this area. That includes faculty within engineering, like my colleague Thanos Panagiotis in chemical and biological engineering, Howard Stone, Miko Hataya, Andre Kosmerl in mechanical and aerospace engineering. I collaborate with José Avalos as well, who we'll hear about. Within molecular biology, there are numerous faculty members, including a really fantastic paper on the way these condensed biomolecular liquids form in metastatic cancer from my colleague Yibin Kang. The list goes on and on. But what I think is really interesting about it is it's a fundamental discovery about the principles by which biomolecules organize within living cells. We can think, immediately we're already doing this. We're pivoting from the fundamental discovery to applications, and that includes the kinds of metabolic engineering applications. We're thinking about therapeutics. There's a lot of interest in the biotech industry on the ways in which drugs might be used to target these types of structures, which are just ubiquitous in cells, dozens and dozens of types of liquid condensed droplets within all of our cells right now. And in many cases, those structures are closely tied to diseases. I mentioned metastatic cancer, Alzheimer's disease, and ALS are thought to be diseases where these liquid condensates actually harden, much like water turns into ice at lower temperatures. So there's a really incredible opportunity for therapeutics and applications, again, even outside of human health. And to go from the fundamental discoveries to these applications is an example of the kind of connection between fundamental and applied research that I think Princeton is maybe the best suited to do. To go from this deep scholarship rooted in intellectual tradition here to thinking about solving problems with that knowledge from our bioengineering efforts.
Yeah, I think your description highlights so nicely so many of the strengths of Princeton in the work that you're talking about. You're talking about collaborating with faculty from many different disciplines, really focused really on the fundamental sciences, on really the fundamental discovery, but that it involves so many of the different disciplines, but then has the application. When you think about the partnerships, their partnerships on campus and their partnerships with entities that are off campus, like I'm sure pharmaceutical companies who might be thinking about therapeutics. Talk to me about how you think about nurturing those partnerships.
Absolutely, yeah. So within the Princeton Bioengineering Initiative, by the way, I'm not sure if we said that, we launched the PBI, the Princeton Bioengineering Initiative, just in November, and we've set up a whole bunch of programs under that initiative. We're really excited about moving forward. We're hiring faculty, we're bringing in postdoctoral scholars, we're setting up new seminar series and so forth. Among those efforts, we're centrally interested in bridging connections and thinking about how we can move the fundamental discoveries that are made here at Princeton into real-world applications. We think that there's an incredible opportunity here in New Jersey, which has a very strong biotech pharmaceutical sector, and we think that bioengineering can tie into and build connections with that industry and those folks. That's being facilitated as well at the much earlier stage of the innovation cycle. In other words, not necessarily licensing technologies and partnering with existing companies, but also with spin-outs. My colleague Rod Priestley, who's the Vice Dean for Innovation at Princeton, we have a lot of ongoing discussions about programs that we'd like to start with Rod's office, including an entrepreneurship program, new coursework around the whole innovation cycle, patents, IP, speaking to venture capitalists, and ultimately spinning out companies. That's something that we would like to have as a cultural feature within bioengineering of innovation, discovery, and application into the real world, and that's going to happen at all these different levels of the innovation cycle.
That's terrific. How do you? I love the visual. How do you think about the partnerships on campus? What are the different disciplines that come together in this bioengineering initiative, and the different departments that it involves on campus?
Well, it's really all across campus. I mean, the Princeton Bioengineering Initiative is based in the School of Engineering and Applied Science. We have a lot of folks within the Chemical and Biological Engineering department, which has nurtured bioengineering on campus for the last 10 plus years. But strong group of faculty members within mechanical and aerospace engineering, strong group within electrical and computer engineering, several within civil and environmental engineering. And within computer science, we have a very strong group of folks using computational approaches to understand the kind of software of the cell. We're going to hear about that from Ben Raphael, who just does amazing work in that area. That's only within engineering. It's across campus within molecular biology. There are many faculty members who are doing really important work, developing new technologies and using engineering approaches to interrogate living systems. That's the other part that we haven't spoken about yet, but living systems are the most complex forms of matter in the universe. There are principles by which they're organized that we have no idea about, we're completely ignorant about. And so the opportunity for learning how these systems work is just tremendous. Again, certainly within human health, but all across society, it's going to have the greatest impact of any discipline. And in order to make this come together, we need folks from chemistry, folks from math, folks from computer science, folks from mechanical and aerospace engineering. And that's what makes this field so exciting. And that's what makes Princeton the best place to take a lead in it.
Fantastic. Well, I'm looking forward to continuing continuing this discussion when you join us a little bit later. I'm now going to turn to talk with Celeste Nelson. But first, let's take a look at some of the work that Assistant Professor of Chemical and Biological Engineering, Sujit Datta, has on a new approach from his lab to track bacterial behavior.
An interesting question is, how do microbes move in a complex, porous environment? And that's a process that influences how diseases spread in your body, how beneficial bacteria can be used to deliver drugs in your body, how beneficial bacteria can be used in environmental settings, how bacteria move through soils to help sustain plant growth. And it turns out that we don't have good answers. Our platform lets us actually figure out how crowding and tight spots constrain how bacteria move. Imagine someone's running around in a ball pit. Those obstacles change how they behave. Our technology is a ball pit for cells, where essentially we use obstructions that we've placed around the cells. We can control the size of those obstructions, how densely packed they are, and how complex their environment is. What our technology relies on is tiny little hydrogel particles that swell up in whatever liquid culture the cells use to survive. As they swell, they form a matrix. Because the system is mainly water, it's transparent. And so we can actually spy on what the individual cells are doing by using fluorescent cells and watching them under the microscope. We can design any microbial community of any 3D architecture and any composition that you want. And what's exciting about that is that we can long-term think about making our own engineered microbial communities to remediate contaminants that are stuck in our soils and sediments and water sources. So that's one example that we're very excited about. Another example is in using beneficial microbes for drug delivery. We have to be able to predict and control how they move through those tight spots and get to where they need to go. And so our platform gives researchers an opportunity to start to answer some of those questions.
We're back with Celeste Nelson, the Wilke Family Professor in Bioengineering and the Director of the Program in Engineering Biology. Welcome, Celeste.
Thanks. Nice to be here with you, Kirsten.
Great. So you and your lab study how organs put themselves together. That's the way I think about it. I wonder if you could give us a more detailed explanation and scientifically sound explanation than my lay understanding of that.
Sure. I mean, that's pretty much what we do. So in the spirit of forward thinking, I have to confess that when I grow up, I aspire to be a tissue engineer. And so I got into this field, really captivated by the idea of regenerative medicine and organ repair and the skills and mindsets that engineers could bring to the problem. But when I got started, I thought, well, maybe we should look to the embryo, which is able to accomplish these amazing feats of engineering organs in order to understand how the embryo builds tissues. And so I looked to developmental biologists and embryologists to help provide answers, to give me a blueprint for how I could help to build organs. And unfortunately found that not enough was known about how the embryo accomplishes these amazing feats of engineering. So in practice, what my lab does is to take engineering and apply it to developmental biology, which is starting to create a new field of developmental bioengineering, which is really focused on understanding the nuts and bolts of how organs and tissues build themselves so that we can pull together nature's toolbox and use those tools to build organs ex vivo or to induce regeneration or repair within the body itself. But when I grow up, my goal is to be a tissue engineer and organ engineer.
So I'm sensing a recurring theme in these conversations that some of the motivation and the drivers is really about the application. And I know certainly regenerative medicine is something that we're very excited about in medicine, because there are so many conditions that we think that being able to really help the body to repair itself and to regrow itself is exactly what we need in order to cure many of the conditions that really plague our society. But you have started with the fundamental roots of this, of how the body has put itself together as it does in the embryo. One of the things that I've been really fascinated when I've heard you describe your work is the people who are in your lab doing this work. Many of them are, in fact, undergraduates.
That's right. And actually, the work that we're doing on my favorite organ, which is the lung, was pioneered in my group by undergraduate students and high school students who bring an energy to the problem and a fearlessness that I really hope to emulate or try to emulate as an older person each time I start a new project. Bioengineering is, as Cliff was saying, one of those fields that students can be really passionate about, and I think of it as a gateway discipline. It's a way to introduce students to engineering concepts and engineering principles, where the applications are so obvious to them that keeps them motivated and helps them bring their skill sets and their ideas to an emerging field that has so many problems that need new viewpoints in order to be solved. That's
wonderful. It feels very distant from my fabulous experience at Princeton in molecular biology, but the engineering quad seemed very distant and not at all related to my everyday experience. Are you actually getting students from different disciplines interested in bioengineering?
Oh, absolutely. If you take a look at the bioengineering labs on campus, it's a smorgasbord of disciplines with respect to the students who are coming in and carrying out the work. But then if you take a step back and look at the students who are taking bioengineering classes, these students come from all four of the divisions on campus. So both from engineering as well as the natural sciences, which are obvious feeders, but also from the humanities and the social sciences. So I've taught classes with classics majors and German majors and history majors and English majors who are really excited about learning about bioengineering and how it touches upon their lives in a way that I wouldn't necessarily have expected when I started teaching here.
Wonderful. Tell us a little bit more about what you're excited about in your lab right now, or either the coolest fundamental discovery or where you think it might be going as you look forward.
These questions always ask us to choose one of our babies. But I think where we've gotten really excited recently is, expanding our view of what's useful to investigate. And what I mean by that is my lab is starting to try to uncover how lungs of different species build themselves. So many organs across the evolutionary tree are quite similar in terms of form that goes along with their function. But the lung is an exception. So across vertebrates, birds, mammals, reptiles have dramatically different lung architectures, despite the fact that the role of this organ is to get oxygen into our bloodstream and to remove carbon dioxide waste products from the blood. And so we've started to investigate the mechanisms that embryos use across different classes of vertebrates to build their lungs. And we're finding just amazing diversity in the types of physical forces and gene expression changes and alterations that take place during building of the lungs. And so we're hopeful that some of the new motifs that we discover can be more easily manipulated and implemented, in a dish to start to build organs outside of the body.
So interesting. Sorry, continue.
Well, so basically we're getting into evolutionary developmental biology, but as engineers, which is another field that I wouldn't have guessed that we would be able to provide contributions to, but we're uncovering new things, and that's exciting to me.
That is very exciting. That is very exciting. Is that true of other organs, or is that particular to the lung, and that it's so different in its architecture across these species?
It seems to be particular to the lung. It's unclear exactly why, but perhaps the why doesn't matter so much as taking advantage of the diversity. And so, yeah, as Cliff was saying, biology is biological systems are some of the most complex machines that you can imagine. And I have this feeling that any developmental motif that we can imagine, evolution has engineered in some way in some species on the planet, either currently or in the past. And so if we can figure out how these tissues do what they do in the embryo, then we'll be better poised to solve some of the pressing challenges related to human health in the future.
That's fantastic. Thank you so much, Celeste. I'm looking forward to continuing this discussion. Next with us, we have José Avalos, just after this break, to hear from promising work from Professors Jean Schwartzbauer and Jeffrey Schwartz with more applications for repairing damaged nerve tissue. We'll be right back.
There are almost 20,000 spinal cord injuries every year in the US. The big challenge is that neurons don't regenerate very well. And so if you have a severe injury in the spinal cord, the neurons aren't going to be able to transit that lesion.
Our technology focuses on creating guidance for this regeneration, and to do that in a way that enables the axons to grow and reconnect in a linear fashion.
So the technology is a combination of chemistry and synthetic polymers with extracellular matrix, which is a biological polymer that controls and organizes all of our tissues, and putting those two together to make different materials that could be used for tissue regeneration in a variety of different human conditions.
Our hope is that using that technology, if axon reconnection occurs, that we will be able to see restoration of function. Spinal cord injury is devastating, and if we could address a few of these, that would make us extremely happy.
I'm glad to welcome to the conversation Assistant Professor of Chemical and Biological Engineering and the Andlinger Center for Energy and the Environment, José Avalos. Welcome, José.
Hi, Kristen. Thank you very much. It's very nice to be with you.
Very nice to have you. So we focused a lot on how bioengineering can help us in human health. Your work really speaks to the potential for bioengineering to help us in all of the many ways in which living systems interact with our human existence. So tell me a little bit about your work.
Right. So this is a very exciting field of bioengineering that oftentimes sort of flies under the radar for many of the people in general in the audience. This is a field of bioengineering that goes beyond sort of biomedical applications and deals more with biomanufacturing, right? The idea of engineering living organisms to use their capabilities to produce all sorts of products that we need for our daily lives, ranging from fuels for energy as well as chemicals to produce everything that we need on a daily basis, to food, to pharmaceuticals, biomaterials. And the idea of this field is to address challenges of global proportions like environmental degradation and sustainability, and ultimately climate change, right? So is the application of bioengineering to dealing with these large, complex issues.
So you're getting the bacteria to do the work for us in a more sustainable way than other ways we think about manufacturing the things that we need.
Correct. In a way, we're actually inheriting the knowledge from millennia, right? We've been using microbes to produce fermented goods like beer and wine and bread and yogurt and all sorts of things. Most of this time without knowing that it was microbes behind the scenes doing all these valuable chemistries. But now with the advent of biotechnology and bioengineering and genetics and genome engineering and so on, we can actually engineer microbes to produce things that they don't normally do. So we can do this by changing their physiologies with what nature already gave them, but also by introducing genes from other species to do chemistries and tasks that they don't naturally do.
So give me an example of what types of things. I mean, I'm all for beer, wine, and bread, and bacteria helping me do that. Tell me about what other things these bacteria, you can engineer them to do.
Sure. So of course, a common example is using these microbes, and they include not only bacteria, they also include yeast, for example, and algae and molds. And so it's, certainly bacteria is a large class of microorganisms that are used, but it's much wider than that. And an example that often comes to mind is that of biofuels, right? We use the applications of, we use algae or other microbes to produce fuels, but we can also use them to produce other chemicals. And for example, in other things that we don't normally think about, for example, food, right? If you think about, for example, a good example is that of farmed salmon. Farmed salmon is white, the meat is white because they don't have their natural diet. But the natural diet is shrimp and other little animals that down up the food chain, right? They assimilate pigments that are naturally produced by algae. And then up the food chain end up in the diet of salmon, of wild salmon, and that's what gives them their pigment. So when you farm them, they don't naturally have this pigment, and so they're white. And so people are not willing to buy white salmon. They probably think that they're being fooled at the supermarket. And so salmon farmers need to dye their fish. And the way they do it today is by feeding them essentially pellets with food with pigments derived from petrochemicals. Now, we could engineer a yeast to produce the very same pigments that the algae makes, so that then we feed them the yeast, which is not only nutritious or has valuable oil, lipids, and vitamins and proteins and so on, but it also has these pigments. And so it would be a more natural way to dye fish, for example.
Very interesting. One of the things that has struck me in hearing about your work is that these microbes could really be engineered to help us across a whole range of applications. Certainly, and probably most compellingly, given the climate crisis, is the biofuels. But talk a little bit about the range of ways in which engineering microbes could actually help with our production.
Sure. So another example that comes to mind that is one of the holy grails in the field is to develop microbes that produce drugs that are very difficult to synthesize and therefore have to be extracted from the natural organisms that make them. So this goes to the heart of sustainability. And so one example is that of Taxol. It's a wonderful drug against all sorts of different kinds of cancer, but it's derived from the bark of a tree, the Pacific yew tree, that takes about 100 years to grow to the size for which you would need to harvest 4 to 6 of these trees to treat one patient. So obviously this is an enormous challenge of sustainability. The trees have been at the brink of extinction, and the cost of these drugs are astronomical. Most people that could benefit from them cannot have access to them because there's simply not enough trees to make the drug. So one of the holy grails in the field is to engineer, say, a yeast that produces these drugs or precursors to these drugs as easily as you grow a batch of beer. Right. So that's sort of the power that this technology can have.
That's incredible. I think when people sometimes hear engineering an organism, they're going to hear, is this related to GMOs, genetically modified organisms? How do you help us can you help us think through GMOs and what does it mean to engineer a living organism to do something different than it usually does?
Sure. So the term GMO is loaded nowadays. And it's really unfortunate because there's a lot of very powerful technologies that can be derived from genetically modified organisms, like the ones I just mentioned, right? Not only for human health, but sustainability, the energy crisis and the climate crisis, right? And so the way that I think of it, there's some legitimate concerns with genetically modified organisms, like releasing them to the environment, right? Especially if they carry some markers that we wouldn't want to propagate, like antibiotic resistance or something like that. The good thing is that there's a lot of safeguards to try to prevent that, especially when a process is going to go into production. And there's a whole field within my field of trying to engineer within these engineered microbes some kill switches, right? So that if they escape, they would not be able to survive or they would commit suicide, right? And so this is the type of technologies that can be integrated into the whole field. Now, there's other less, so in my mind, the biggest concern is the release into the environment because of the potential dangers of polluting the environment with these types of organisms. Less of a concern are issues that are more often in the public, like eating, for example, a salmon that was dyed with yeast that was engineered with the same gene as this algae to make it more sustainable and actually healthier, because then you're dyeing it with the actual pigment that naturally dyes the salmon, as opposed to with a petrochemical, right? So unfortunately there's a lot of misinformation and misunderstanding in the public that confuse these two different types of dangers, one being legitimate, the other one not so much.
Yeah, I like that you've really hit on also the dangers in many of the chemicals that we've synthesized in some other way, and I really like the example that you gave of being thoughtful about how you can think in engineering the organisms to produce, that you can also think through the safeguards at the same time, and sustainability is so much of a key here. I think that's what's so fascinating about your work, especially in the context of all of our discussions around the need to think through a sustainable planet and a sustainable way of life. So thank you very much. I'm looking forward to having you join us again. So next we'll speak with Ben Raphael about bioengineering from the perspective of a computer scientist. But first, we get a glimpse at a protein targeting technique from the James S. McDonald Distinguished University Professor of Chemistry, David McMillan. David and his colleagues call this technology micromapping, which is some pretty forward-thinking applications, even potentially helping to find antiviral treatments for COVID-19. Let's take a look.
What we've invented is a technology we call micromapping. And the idea behind it is that either within a cell or on the surface of a cell, it would be really valuable to be able to understand what is in the neighborhood of any given biological target. The key ingredient to all this technology is light, and light is a wonderful ingredient because light, as we know, you can switch it on, switch it off, and blue light is very much benign to cells. So you might take a cell and take a snapshot at the beginning, but then you might do something to the cell and see which proteins change. Not only can we understand what the cell's doing, we can actually follow it in real time to see how it's evolving. And the second thing is light can obviously get to the surface of cells and it can penetrate into cells. So we're using these new photocatalysts and light that allow you to label proteins to see what's all around these particular systems in a way that we just couldn't do before. The disease states that we're working on is numerous areas of oncology. We're also working on a range of neurological diseases, including Alzheimer's And we're also working in viral diseases. Another application is in the area of COVID. And what we found is that we can see not only where the spike protein interacts, where we know that the virus is basically pulled in to normal healthy cells, but we've actually now started to find lots of other information about what the virus is doing, which helps it become effectively absorbed into cells. But that's just one thin slice of one of the things we can do with it. The number one thing is to get it in as many people's hands as fast as possible, because the insights that will come out of it will drive science in so many different areas. From our point of view, that's been very exciting.
Although Ben Raphael is a professor of computer science, he develops and applies computational and mathematical methods to identify and address complex biological processes. Thanks, Ben, for being with us today to share your perspective.
Thank you, Kirsten. Great to be here.
So I know when we prepped for this, you were first to tell me that you're not a bioengineer. So tell me why a computer scientist is relevant for this bioengineering forward-thinking Forward Fest.
Yes, well, it's a great question. And when we think about bioengineering, these are two very broad disciplines, right? Biology is extremely broad discipline, engineering an extremely broad discipline, and both disciplines with many departments, many subspecialties. So I represent, I guess, one slice of that intersection between biology and engineering, and that's the intersection of computer science and molecular biology primarily. And it's a fascinating intersection to be at. It's a very exciting time to be working in this area. In my group, we use techniques from computer science and machine learning and statistics and mathematics to interpret primarily molecular data. So that's high throughput measurements of DNA, RNA, and proteins, and mostly measurements that are made in human and cancer samples. It's a really amazing time to be working in bioengineering. It's a really amazing time to be working in computational biology. So maybe just to give you kind of one example of that from cancer genomics, last month was the 20th anniversary of the publication of the human genome. It's a project that completed a single human reference genome and cost about $3 billion to do that. A few years ago, I was part of a large project called the Cancer Genome Atlas that sequenced genomes from 10,000 cancer patients from 33 different cancer types. So you see the sort of scale of things growing. And a few months ago, we published a paper where we analyzed DNA sequencing data from 10,000 individual cells from a single breast cancer patient. And by going in that level of depth, one can sort of analyze the mutations that are present even within the different cells of a single tumor, and how these different mutations interact with each other and allow the tumor to evolve in response to treatment. So, this acceleration of the volume of biological data that's being produced really doesn't show any signs of slowing down. So, it's a great time to be a computational biologist 20 years ago when the first human genome was being sequenced. It's an amazing time to be a computational biologist now, and it's going to be a phenomenal time to be a computational biologist, tomorrow or 5 years from now or 10 or 20 years from now.
Yeah, it's amazing when you think through the applications of our genetic technologies to understanding cancer, but to think about each cell within a tumor and having mapped all of that and having these extraordinary computational tools that allow you to give meaning to all of that mapping is really extraordinary. And the way I think about your work, and tell me if I'm wrong, I can really then start to understand what happens to a tumor over time, even, or how a metastatic cancer might develop from a tumor. Tell us a little bit more about how we should think through the applications of this extraordinary computational power.
Yeah, so most cancers are driven, primarily, but not exclusively, but primarily by mutations and other molecular changes within the cells of the patient. And so understanding what those changes are is extremely important for trying to develop treatments that are targeted towards the particular subtype of cancer that the patient has. I mean, cancer, we talk about it as a disease, but it's really, tens of thousands of diseases when you think about all the complexity that is there. And we really need to think of it that way. We shouldn't think about cancer. We shouldn't think about lung cancer or breast cancer as sort of monolithic diseases. They're really very, very diverse, heterogeneous, and we really would like to understand that heterogeneity. Now, if every tumor was completely different and somehow completely random, it would be really difficult to imagine how we might develop any reasonable treatments. But there's fortunately for us, in terms of thinking about treatment, the mutations that occur in cancer patients, although they might be quite diverse, actually seemingly converge on a small number of biological processes, right? So cancers need to grow quickly. They want to evade the immune system. They'd like to metastasize. And each of those biological processes has some molecular circuitry associated with it. So one of the things we've tried to do is understand the mapping from genotype to the mutations to the phenotype, to what are these molecular processes that are being perturbed, and how can we understand what they are at the level of groups of patients, but really at the level of an individual patient.
Right. So that one of the challenges, of course, in cancer is that our still our primary mode of dealing with cancer is to give people essentially poisons, and figuring out how we can more assuredly give it the right treatment to the right patient at the right time. It really, I think, relies on understanding the tumors, and it sounds like this is the type of information that will really help us to do that better.
Yes, and I mean it's changing very rapidly. It's really amazing the pace of change that there's been. I mean, we're still using poisons, chemotherapies routinely, but in most major cancer centers now, if a patient goes in, they'll have at a minimum a few hundred genes in their cancer that are sequenced, genes that are known to have mutations that are clinically relevant. Some cancer centers might now be sequencing all the genes in the genome of cancer patients and try to identify even rarer mutations that might be important. We're even getting to the point where we're sequencing the whole genomes of cancer cells, and ultimately maybe even sequence many cells within a tumor to understand this sort of evolving ecosystem that makes up the tumor. So it's moving quickly. And in addition to just mutations, I mean, the other angle that's been amazing is to really think about how cancers and the immune system interact. And that's been just a huge rapid development in terms of the transformative nature of immunotherapies. And both of those things together are just really accelerating.
Tell me, how far are we away from, so you're publishing this atlas and helping me to understand every cell within a tumor. Can we imagine a time when I'm actually with the oncologist doing these types of calculations, the types of analyses for an individual patient more in real time to guide decisions in real time?
Yeah, I mean, it's happening a bit now. I mean, there are in many cancer centers, they've routinely run the tumor board. They now have the molecular tumor board, where in addition to talking about all the usual clinical variables, they talk about what happened in the sequencing and what mutations were in that genome and what treatments might be that patient might be responsive to based on their mutational profile. So it's already happening. I think one of the challenges that one thinks about is that the volume of information is growing so readily that there'll be a point where the individual oncologist or group of oncologists probably can't keep all of the latest data in their head. And so what's needed is, again, to bring the power of machine learning right into the oncologist office, right? So they could take whatever amazing high throughput measurements they have of the tumor and really get sort of a much more focused view of what might be the most relevant things for that patient.
So I'm going to ask you the question I asked Cliff. So, I'm at a medical school and our oncologists are really excited about it. You're giving me all these cancer applications. You're not at a medical school. So tell me about the collaborations that you have and why Princeton is the right place to do this. Yeah,
well, I mean, we get data from lots of different collaborations, medical schools all over the country, all over the world, small collaborations with individual investigators, large international collaborations. We're fortunate that in the genomics world, there's a long history of data sharing. So we're able to get access to this data. And one of the things that I think is really exciting about Princeton, the environment, is it's just such an outstanding research environment, just really stellar students in all areas. But, in my view, for computer science and machine learning and all these types of things. And so we're able to, leverage the strength of the Princeton environment when we analyze data, and then we can bring things back in our things we might identify in these large data sets and have interactions with, Cliff's lab or Celeste's lab or, many other labs in throughout campus to try to really dig deep into one of the discoveries that we're finding in these large data sets. So, the sort of, external collaborations, but internal collaborations in this really rich environment, I think is very distinctive at Princeton.
Great, but we'll talk about that more when we bring all of you back together. But thanks so much. And please stay where you are, because after this break, we will bring all of today's forward thinkers back for a lively discussion. While they get ready to come back, let's hear from Esteban Engel, researcher in viral neuroengineering in the Princeton Neuroscience Institute, whose team has developed a system for switching on the activities of genes that can improve treatments for a broad range of neurological diseases.
As the global population is aging, there's an increasing number of neurodegenerative diseases that remain untreated. The technology we have developed can be used to deliver genes to all neurons in the nervous system. We just piggyback on a virus to deliver new information into cells. What we innovated is to use a fragment of DNA called gene promoter that is much smaller than the promoters being used today. In some diseases, the gene that has to be fixed is large in size, and with the currently used gene promoters that are also quite large, it was not possible to include that into the payload. With our engineered viruses, the promoter is so small that there's more room, and this allows us to deliver larger genes or multiple small genes. The problem with promoters, regardless of their size, is they shut off after a few weeks or a few months. And when you're giving a gene therapy, you want that to be long-lasting. With our engineered viruses, genes can remain active for as long as they're needed.
I'm joined now by all four of our speakers. And before we get to the questions from the audience, I want to start with a few questions that we can each bring us back to some of the themes we talked about in many of our discussions with each of you individually. So one of the things that you've made it clear why Princeton is exactly the right place to be doing this type of cross-disciplinary, interdisciplinary work. I wonder if you could also speak to what it is about Princeton as a liberal arts institution that really makes it the right environment to think about this very forward-thinking field of bioengineering. I'll throw it open to whoever wants to start.
I could make a comment if you like, Kirsten.
Yeah.
Yeah, I would first say with any new field, new technologies, there's always going to be incredible, societal discourse, and there will be ethical dimensions around it. We're seeing that, of course, now with the digital revolution and questions of privacy and equity and all these things that emerge from the widespread adoption of, everybody has a computer in their pocket now. That's certainly happening in bioengineering. We touched a little bit upon that in genetically modified organisms. That discussion, although I would say certainly people have strong opinions about it, is going to pale in importance to the discussion that is emerging around the CRISPR technologies, the ability to engineer the genome and engineer, as people say, the germline, which will have an unprecedented impact on the evolution of humanity. I mean, people have said that, if you took a human from 1,000 years ago and you clean them up and dress them up, you wouldn't tell the difference. But if you took a human from 1,000 years in the future and brought them to today, you might very well know that they're different. So that's something that is going to be incredible ethical dimensions to this discussion, which is already emerging. And it's critical that this is done in a deliberative manner with input from policy makers, from discussions of, historians, philosophers, all aspects. And that, I think, can only happen at a place where these different disciplines coexist. And we've discussed the bioengineering and embrace of engineering within the liberal arts education of Princeton. And I think this is a perfect place to take bioengineering in the right direction that it should go, and to think carefully about the implications. And there's no question that there will be some incredibly powerful technologies that have absolutely game-changing impact on society. We have to do this in a careful way, and Princeton's a great place to do it.
It strikes me that Celeste, what you were saying, that you're able to bring in so many people from other undergraduates from so many other disciplines as sort of a gateway into this. It strikes me that this is a great environment, particularly if bioengineering as a field that can actually draw others into it, who might be also then helping us to understand the context of these new discoveries and their applications.
Absolutely. I mean, I think that, sometimes we tend to get a little bit, siloed in our own fields and start thinking about the applications and the implications, from the perspective of that field alone. But when we, start to explain what we're doing or the aspirations that we have for the future to people who come at these problems from very different backgrounds, they can raise issues related to ethics and applications and implications that we might not, see on a daily basis. And I've certainly experienced those conversations in the classroom when you have, students majoring in a variety of different disciplines talking about CRISPR or CRISPR-edited babies or genetically modified organisms. And it increases the richness of the discussion, but also the richness that we bring back to our own labs. So Princeton's outstanding for that.
Great. So it's a two-way discussion or a multi-angle discussion, not just people commenting on bioengineering, but very good. Very good.
Yeah,
anyone else want to come? Yeah, go ahead.
I was going to add one point. I mean, to Celeste's point about, things being siloed. I mean, in a young field, an interdisciplinary field like bioengineering, you don't want it to become siloed too early. You want these diverse intellectual viewpoints colliding with each other, routinely, so that, the field is continued to be enriched, and you don't want to define what it is early. And one nice thing about Princeton is that it's a relatively small place where people collide into each other routinely. And so, it's hard. It's almost impossible to become siloed at Princeton. You're sort of forced to, interact with the different disciplines and explain your work, hear their viewpoint, and mutually sort of reinforcing these things. And I think that's critical in a new field. It's critical in an interdisciplinary field, critical in a field where, biology and engineering have for a long time been separated. When you sort of bring these diverse viewpoints together, you want that to stay an open conversation for, time into the future.
Great. I have another question from one of our audience members, and we've talked a lot about partnerships and certainly about the applications of many of the discoveries that are happening in bioengineering. How do we as an institution, how do you as scientists think about the motivations of industry, and how do we balance the ethos in the academy with the motivations of industry when we are trying to really bring discovery to its application? And how do we think about the collaborations and who's really running the show in these collaborations, and where are the guardrails to ensure that the principles that we hold dear at Princeton are really maintained, while making sure we actually reach the applications to human health and sustainability of the environment? Industry collaborations. Who wants to jump in?
Well, I can just say briefly that I feel that Princeton really values the work of their researchers. And I feel when I've had conversations around this topic, this question, I always feel that that Princeton has sort of built in some of these safeguards, as you say, to make sure that there's still intellectual freedom for the researcher to pursue the fundamental questions or develop the technologies that they see as the most important and valuable for their research, as opposed to becoming sort of just subservient to some other form of funding. And so this encourages to have these very open discussion up front, so that any collaboration with industry is based on the understanding of this intellectual freedom, the ability to publish, the ability to patent, and so on. So I have not actually encountered this particular problem.
Terrific. Anyone else want to comment?
I think the word that Jose used that
rings the most true is values. I think Princeton is a campus that values fundamental discovery and basic discovery, basic science. And the applications are fantastic and incredibly important, and many of us are motivated by them. But when it comes down to it, we're a campus of people who are excited about how the world works. Mm-hmm. And bioengineers on this campus are no different. And so if that is your intrinsic value, then any collaboration is going to be shaped by that.
Cliff, I think that's so well said and resonates for me with how Princeton exists and certainly how I imagine it would be to be a scientist at Princeton. Cliff, one of the things that has been part of the bioengineering initiative has really been also to talk about the entrepreneurship of our own faculty. Talk to me about how you look at the efforts to really catalyze that and how do we balance that amongst our faculty?
Yeah, I mean, I think it's a really important question. For us, we view the translation of the basic discoveries into the real world as, in some sense, this is where the sort of rubber hits the road in terms of the impact on the world. That doesn't mean that we're not motivated by fundamental discovery. I mean, I tell people that a lot. I mean, the main thing that gets me up in the morning is the thought that, wow, we could really learn something new and potentially be the first, human to learn how something works. That to me is the, this idea of fundamental discovery and applications, technologies that we develop that have application that industries become interested in. I think you have to proceed in a thoughtful way about it. I mean, we don't want to, as Jose said, have some kind of a situation where people feel like, oh, they're really working for some company or things like that. But honestly, I feel that there's no such danger. I mean, in my experience with industry collaborations and my recent experience in spinning out a company from our lab, the university already has in place a whole bunch of safeguards. The bottom line at the end of the day is no relationship, no formal relationship with any external entity will be allowed that restricts your freedom to operate in intellectual endeavors and however you see fit. I mean, that's the mission of the university, right? We should be able to say, hey, I want to study this, I want to do this, I want to, open up a new area that combines this and this. So the other part of it I would say is that a lot of this is driven by students, right? Students are deeply interested in entrepreneurship and connections with industry. Many of our students go off into industry. So I think, we can't pretend that there's not this commercial world out there where, that's where products are made. And that's where, look at the vaccines that most of us, at least me, are eagerly waiting to get. I mean, these are commercial enterprises and we need that. That's how, that's how society works. But certainly there should be a conversation about how best to do that in no way compromises scholarship. And that's absolutely the top priority.
Great. So, Jose, you said that your subdiscipline sometimes flies under the radar. And I think Ben has also mentioned that bioengineering as a whole is a relatively new field. So as the mainstream media starts to talk about this field, what is the one thing from your viewpoint that you'd like the general public to be made aware of with regard to this field or your specialty so that they can be knowledgeable when they're taking in the news or the new developments as they emerge in the mainstream media? I'm going to start with Jose, if that's okay. And then, but I'd love each of you to say, what can we each do to understand your field better and be consumers once the mainstream press gets a hold of it?
Sure. So what I mean by flying under the radar is because when you hear people talking about biotech, the first thing that comes to mind usually is some biomedical application, and that's fair because it impacts everyone most directly, I would say. However, there's this whole field of bioengineering that does not have to do directly with biomedical applications, but rather with what has been called like the future bioeconomy. So one way to think about it is right now, our society is driven by petroleum, right? We live in an oil based economy. In the future, that's going to change. It's going to be a bio-based economy. So hence the term bioeconomy, in which most of the products and goods that we enjoy that are currently either directly or indirectly derived from petroleum will be derived from bioproducts, from bioengineering. And so when people are, as Cliff said, every time there's a new technology, there's a discussion, there's skepticism, there's concerns and questions. I would suggest people ask themselves whether the new technology is any more dangerous than, for example, the current petrochemicals that we use, and hence this example that I thought about with the salmon, right? Is it really safer to eat a fish that is dyed with a petrochemical as opposed to one that is dyed with the actual pigment that naturally dyes the fish? And so I would say that it would be very important for the society to be well informed and sort of embrace the aspects, the good aspects of bioengineering, everything it has to offer, of course, keeping in mind to do all of these advances safely.
Great, thank you. Who else wants to tell us the one thing they'd like somebody to know? That could help us to be better consumers of the media as consumers of these new discoveries as they emerge in bioengineering.
Yeah, I could just make a comment on that, Kirsten. I mean, I think it's our responsibility as scientists and engineers and researchers to communicate well the ideas and to better communicate them. I think, frankly, historically, I believe there hasn't been as much of an effort to, we sort of speak this esoteric language because we've been speaking it for decades, and we don't really try that hard to communicate to people that aren't in on the jargon and so forth. I think we could all do a better job of that. At the same time, I do think that, as these technologies, they're clearly transforming society. I mean, the fact that we're having this meeting digitally and everybody's working from home, and now the real estate market's crazy and the economy does, it has, obviously that's related to COVID, but our ability to handle it in this particular way, I just think that society needs to understand at a broad level how these technologies work. I also think that it's an essential thing for a functional society. You see all these conspiracy theories, people saying that there's microchips in the vaccines and this sort of thing. I've had conversations with folks and pointed out that technology doesn't yet exist, so you don't have to worry about that. But these are things, and we need an informed society, and it's our responsibility. But I would say at the same time, I think people need to try to educate themselves about even the basics about how these technologies work.
Great, Ben. What would you like us to know?
Yeah, I mean, echoing what Cliff and Jose said, I mean, I think, massive potential in how we diagnose and treat disease. But at the same time, lots of things to be cautious of. It's often a hard road from the biological discovery to the translation into patients. It's unpredictable sometimes which of those discoveries are going to bear fruit and which will not. And so, I think there's lots of excitement. There should be tremendous promise. We're going to see tremendous progress, I think. But, it's not going to be just a straight up, linear path. There's, going to be lots of things that are, don't work, don't fulfill their promise. Others that we couldn't have predicted that are going to work amazingly well. And, if you think about what happened with the coronavirus vaccines, and, these were the very first vaccines that used RNA technology, a technology that was like sitting in kind of the backwater for a couple of decades, never quite working. And then amazingly within, months, just has had a transformative impact on society. And I think we'll see a lot of things like that. There'll be these technologies that have this transformative impact that, might have, been around in the lab for 15 or 20 years before suddenly it's right place, right time, and they reach their potential. So tremendous promise, but also I think, the cautions that Jose and Cliff brought up around, safety and what are the guardrails that we want as a society? How do we want to best use these technologies? I mean, certainly we, like to treat and cure diseases, prolong life, make for better life. Do we, want to, start editing embryos? I mean, these are going to be thorny questions that as a society we're going to have to start addressing as these technologies allow us to do a lot more than has ever been possible in human history.
Yeah, it's so well said and so relevant to what you're talking about and the work that you are all doing at Princeton, which is so much about the fundamental discoveries which have the potential for future, but it's sometimes the timeline is we don't yet know the timeline to the applications, and the mRNA vaccines is such a great example. Celeste, any thoughts of what you would like us to know as we continue to watch the exciting discoveries coming from the field of bioengineering?
I think, just echoing what's been said so far, that we as bioengineers probably need to do a better job of articulating the timelines and how long many of these applications take. I mean, the COVID vaccine appears to have, been a huge success story in about a year, but as Ben said, the fundamental technology that many of the products rely on go back decades. I mentioned at the beginning when we were chatting individually that I aspire to be a tissue engineer, and this is a branch of bioengineering that came out with a lot of hype in the 1990s, and probably overstated some of the promise and the timelines to its detriment. And I feel like we need to be a little bit cautious in being circumspect about how long things will take and how challenging many of the applications really are, while at the same time highlighting how exciting the discoveries that we're making are to understanding who we are as human beings and how we fit into the planet and what we can do to make our lives and society better.
Yeah, that's really a fantastic note to finish this wonderful discussion on. I'm really looking forward to watching and celebrating each of your discoveries and those of Princeton Bioengineering Initiative. I thank you for joining us here today, and thank everyone in the audience for this really fantastic discussion. Thank you very much.
Thank you for engaging in this Forward Fest. Be sure to keep the conversation going on social media with #PrincetonForward, and visit forwardthinking.princeton.edu to access more forward-thinking content, including a downloadable resource guide for this month's Forward Fest that will help you dive deeper into these ideas. Again, we're so glad you've been a part of Forward Fest. Until next time, keep up the forward thinking.